Hi, thank you. Thank you for the introduction. So I wasn't sure exactly how to um, gauge this talk, and I thought I would give sort of like an introductory talk about genome architecture and sort of how we use experimental data and how we use that data to make theoretical models and how we confirm these models. So, but it's fairly general introduction, I would say. So we won't go in all the details of the modeling or the computation or the experiments. So my lab is interested in genome architecture and is interested in genome architecture from the point of view of theory, which is also one of the major directions of the Center for Theoretical Biology of Physics. So the first thing to notice is our genome is very long, and human genome is about 2 meters long. It's 46 molecules. The longest of each one of these molecules, the longest of our chromosomes, is about 30 centimeters long. And this genome is stored in the nucleus, which is very small. It's about 6 microns across. And typically, when we think about the genome, we think about its function as a storage device. You know, the genome is storing the sequences of the proteins, and then the proteins they do various things in cells. So this is traditional view, and is essentially a central dogma of molecular biology, and as many other dogmas, is at least partially incorrect, in the sense that a key feature of this dogma is that the flow of information goes from DNA to protein and there's never a feedback. And we know that that's not true. We know that there is epigenetics, and epigenetics is sort of writing down a different information on the DNA using the post-translational modification on the histone tails. This signal is different from the DNA signal in the sense that it can be written during the life of the cell, and it can modify the way the genome is translated. And by the way, we have very effective ways to read the epigenetic sequences from DNA, and those are mainly ChIP-seq experiments, chromatin immunoprecipitation and sequencing. And these experiments, they report that tells us how much of a certain post-translational modification is found at a log of some DNA. Okay, so arising from that, from the fact that we have a genome and there's DNA and there's epigenetics, and we're starting to understand in the last, I'd say, last 2 decades, that DNA is more than a storage device. DNA is not just a tape on which the sequences of protein are written on, but DNA really participate into the information processing of a cell. So in this figure, which is just just an example, you have an inflammatory stimulus. The inflammatory stimulus is get read inside the cell by a chain of protein-protein interactions, and then these signals arrives to the nucleus, and in the nucleus there's some additional processing of this information to then some genes are activated, some proteins are produced, and then they participate to the function of the cells. And that's why in the logo of my lab, I actually put the proteins like these little gears, and there's a feedback between DNA translation and production of protein participate. So overall, it becomes clear that the genome is an ensemble of interacting macromolecules that participate in information processing, and part of this information processing goes through structure. In protein, we have this strong connection between structure and function. In the genome, this connection is probable, but it has to be studied, and this is sort of like one of the topics of research in my lab. We know for hundreds of years that the structure of the genome changes to adapt to the function. For example, we know that the structure of the genome changes along the cell cycle, going from, let me see if I can find a pointer. That's fine. I think I will. Where's the pointer? Okay, here it is. So we have the iconic shape of chromosomes, which is found in metaphase, while in interphase the chromosomes are open and accessible for translation. So this happens along the cell cycle, but also there are changes in the structure that happen with differentiation. So we now know that the structure of differentiated cells is different. So we have the same DNA that's some different conformations in different tissues. So cells that perform different functions. And as well, the conformation of the genome changes along evolution. So most or a great part of what we know about the organization of the genome is due to these techniques that have been developed in the past 15, 20 years, which are DNA proximity ligations. In these techniques, what happens is that DNA is cross-linked. Here we have two strands of DNA, a blue and an orange, and these two strands of DNA, they happen to be in close physical proximity, therefore are cross-linked. Then there are cut restriction enzyme, the open ends of DNA, they are ligated, and then purified and sheared to produce a library of these sequences in which we have a blue piece and an orange piece. And these chimera sequences, you know, they are pieces of DNA that happen to be in close physical proximity, but maybe far away along the polymer. Then we read all of these sequences and we create a contact frequency map, as we can see here, which the orange bit of DNA will align to a logos I, and the blue bit of DNA will align to a logos J. Then we repeat this for a few billion of contacts, and then we have this contact frequency maps. So what did we learn from these contact frequency maps? We learned that the structure of the genome is conserved, and it's cell type specific. So here we see a few human cell lines. We see that the structure is conserved. There's these domains along the diagonal, which are called topological associated domains. Here we see the same piece of DNA, which is found in chromosome 14 in human and in chromosome 12 in mouse. This is the same genes. And we find that the same genes that have the same conformations in human and mouse, indicating that that sort of structural organization of the genome is conserved along evolution. So a few years ago, we started from observing this data. We set up on a journey to define energy landscape theory for chromatin organization. And the energy landscape theory that we defined through with our work through the years, it goes in two steps. There's the two steps that carry us from a one-dimensional information about epigenetic information, which comes in the form of ChIP-seq information, all the way to an ensemble of three-dimensional structure for that chromosome. And the intermediate step is a step in which the sequence of epigenetic information is converted into a sequence of chromatin structural types. So this was sort of the overarching ansatz that we made a few years ago. And the idea is to mimic what was the thinking for protein. In protein, you have a sequence of amino acids, and the sequence of amino acids determine the 3D structure of the protein. And the answer was that there's something similar in chromosomes. There's a sequence of chromatin structural types, and the sequence of chromatin structural types determines the three-dimensional fold of the chromosome. Now, the sequence of chromatin structural types is due to epigenetic information, to epigenetic markings. So it's not related to the DNA. So it can be dynamically rewritten to accommodate different folds in different chromosomes, in different cell types. So that was the assumption. And this assumption is related, it turns out that in physics terms, it's the idea that there is a phase separation of chromatin structural types. If and when two segments of chromatin form a contact, the energy of that contact depends on the type identity of that contact. This ansatz, this physical ansatz, was due by, was inspired by observing the experimental data. In these Heisey maps that you see here, this is a region far away from the diagonal, and in this region we see a typical feature of Heisey maps, which is compartmentalization. It's this checkerboard pattern that we observe here. So when we look into this pattern of interactions, we see that there are not an infinite number of patterns, but we see that the patterns they repeat. For example, this pattern here, which is labeled by the purple color, is characterized by this light, dark, light, dark, light, dark pattern, and it's the same, for example, in this other region, which is also labeled as purple. So looking at the Heisey maps, we can do this labeling in colors, and each colors will correspond to a chromatin structural types or a pattern of interactions. Of such patterns, in this human cell line, we found 6. And this pattern correlates very well with the epigenetic information you can see here in this ChIP-seq tracks. So for example, each purple bit of chromatin happened to have this H4K20 trimethylation enhanced with respect to other chromatin structural types. And here you can see actually on the right-hand side, you have 2 human cell lines, and the structure of these 2 human cell lines is different, as you can see from the Heisey map, in which you have a dark region and a light region, or on IMR90, you have light, dark, light, dark. So the structure is different, and associated with this change in structure, there's a change in H3K36 methylation, which you see in the green ChIP-seq track. So this is the same DNA, different structure, and different epigenetic markings. So that's very clear connection between these 2 things. So this was the first sort of physical process that shapes the conformation of chromosomes. The second physical process that shapes chromosome is lengthwise compaction, which at the time we sort of found it in Heisey data we call the ideal chromosome. And this energy term, it says that every time two loci at the genomic distance d come into contact, there's a gain of, or a loss of, an effective free energy gamma that depend on the genomic distance interspacing these 2 loci making a contact. So this is a term that in the modeling, in sort of the energy, in the energy function of proteins, it's been, for example, could be a term that give rise to alpha helix. Alpha helix, it's a preferential contact formed between an amino acid at position I and an amino acid at position I+4. In this case, it's not indicating, it's not due to the chemical identity of the DNA or the epigenetic modification, but we believe this is actually the effect of protein motors that act along DNA. After we found this energy term, we called it the ideal chromosome, then DNA extrusion has been discovered. And in DNA extrusion, we have ring-shaped protein complexes dock over DNA and extrude loops that are largest. And and there's also other proteins that act on the polymer, on the DNA polymer. We have topoisomerases, we have RNA polymerase, and possibly other unknown motor activity. But the fact is, the effect of all of these motors acting along the polymer generate an effective free energy, and this effective free energy might depend on the genomic distance, or in terms of the protein motors, that might depend on how long these motors they reside on DNA. So using this sort of two simple and guesses about the physical processes, we could write an energy function, which I call the minimal chromatin model. And this energy function has a polymer potential, has type-to-type interactions, and it has this ideal chromosome term. So again, the type-to-type interaction act exclusively across space. There's no dependence on genomic distance, while the ideal chromosome, there's a dependence on Cartesian distance, but there's also dependence on genomic distance to include effects that act along the chromatin fiber. So the second step that would allow us, so this energy function can be used in molecular dynamic simulations once it's been trained. But the other ingredient that we need to sort of like predict the co-formation of chromosomes is to figure out what is the sequence of chromatin structural types, which we do with a machine learning step, which we call megabase. Megabase, essentially, what we did is we aligned the compartment patterns that are observed in high C with the ChIP-seq formation for that same line, for same cell line, and then we create a database in which for each locus we write down the type, the observed chromatin structural types, and all the experimental values at the location of the different epigenetic modification. For many loci, we write down the sequences, we build a database of such sequences, then using a Hamiltonian of a spin glass or a Potts model, we use that Hamiltonian to infer which one is the most likely chromatin structural type given the sequence of epigenetic modification observed at the locus. Now, with that, we have a full pipeline. We go on a freely accessible database from NIH, like the ENCODE database. We read the epigenetic modification for the chromosome we're interested into and the cell lines we're interested into. Then we run megabase to produce compartment annotation or chromatin structural types for that chromosome, which is a single sequence of letters. Then we run molecular dynamics, and the molecular dynamics returns us an ensemble of three-dimensional configuration for that chromosome. And then we can create in silico high C, and then we can go compare that high C with the experimental high C, and we can see that we can, sort of like validate our work. And the bottom line is that the simulated, the predicted structural ensembles are very, very similar to the experimental ensembles. Here you can see the patterns of compartmentalization were reproduced by the simulations are here on top, and the experiments are on bottom. The two maps are very, very well correlated. The compartmentalization patterns are very well reproduced. And this is for a single human cell line. And then we started looking in different human cell lines, like can we predict the conformation of all human cell lines? And here you can see 3 human cell lines, for example, and it's the same chromosome, it's chromosome 21, and you can see that the structures are different. Here you have, as usual, you have experiment on the bottom part of the map and simulation on the top part of the map. And you can see that there are differences between the 3 cell lines. So this region here, it's different from a lymphoblastoid to epithelial and endothelial cells. And you can see that the simulation well predict that difference. And then we had, for this, we have performed simulation for tens of human cell lines. For each cell line, we do all the chromosomes. And we're working with the ENCODE consortium to build an atlas of structural ensembles for all human tissues and cell lines. By that time, we had data for tens of cell lines and hundreds of chromosomes. So essentially, we have, for each cell line, we have about 500,000 individual. For each chromosome of each cell line, we have about 500,000 individual structures. So we had hundreds of millions of individual structures for chromosomes. And we wanted to share all this data, structural data, and also the methods that we used to calculate these conformations or to analyze them. We wanted to share with the community of researchers. So we created this tool, this data bank. It's called the Nucleome Data Bank, which is at ndb.rice.edu. And there you can find all the simulations that we ever run. You can find you can run from the server Megabase, you can run from the server Micron. And we're working also to integrate this these tools with the output of imaging experiments that produce sort of the similar set of data, which are 3D conformations. So a couple of things. One thing was interesting at the beginning was that the results of our simulations were slightly different from what other group were seeing, in the sense that we were observing very dynamical organization of chromosomes. Chromosomes are very fluid. There's not a single conformation like in a protein. There's not a native structure, but it's more of like chemical, these propensities that are created by the proteins and the epigenetic modification that creates this sort of microphase separation, which is very dynamic, in which droplets splits and fuse. And so this was a very clear outcome of our modeling. And so we tried to figure out, if this was corresponding to what experiments testing the dynamics of chromosomes in vivo were observing. And so we started looking at imaging data, fish data. We could reproduce the fish data. Then more recently, there's DNA tracing experiments that involve individual 3D structures. So we could sort of calculate the radius of gyration of our structures and the heterogeneity of our structures and compare it with the heterogeneity of single cell imaging. And we found very good agreement. And then we looked at the dynamics experiments, optical experiments probing chromosomal dynamics. They were seeing subdiffusivity of loci. We're seeing the viscoelasticity, which is evident by this sort of negative peak in the velocity autocorrelation function. And they're seeing a spatial coherence. And all of these three sort of separate observation, they sort of like match in our model. So we see subdiffusivity with the exponent which is similar to what's been observed in experiment of about 0.3. We see the viscoelasticity, and we see the coherence of the motion of loci, which is seen in correlation spectroscopy experiments. Another sort of unexpected feature of our structural ensembles is that they don't have a lot of knots, and this was sort of like assumed in posterior, because if there is a lot of knots in chromosomes, then you expect that it's going to be difficult to separate the chromosomes during mitosis. That was sort of like a design requirement, but it wasn't clear how this absence of knots was achieved. So we just looked into our structures, and we observed that the structures coming from the minimal chromatin model have a lot less knots than a homopolymer of a similar length, which you can see here in the two distributions in blue and red. So these distributions, they indicate one specific measure of knotedness, which is the minimum rope length. And very simply, the minimum rope length is the length of a rope you need to make a knot of certain complexity. And of course, the more complex is the knot, the longer is the rope you need for that to make that knot. And so if you start with a homopolymer, well, homopolymer is naturally knotted, and that's sort of like an equilibrium globule. If you take a homopolymer, you let it equilibrate, you get an ensemble of structures which are fairly knotted. Then what we did was we turned on our minimal chromatin model potential, and that's effectively removing knots. So it equilibrates because we allow for chain crossing, which is sort of like what you should do, because that's what happens in the nucleus because of the activity of topoisomerase protein. So the action of that potential is to remove knots, to go down to an equilibrium structure, which has no knots or very little knotting. And it's the equilibrium state of this lengthwise compacted polymer, as we have explained, and phase separated polymer. And that's sort of like an alternative explanation to the model that has been around for a number of years, which is a fractal globule, in which you have that the chromosome, it's a homopolymer, but it never reaches equilibrium. So it's long-lived non-equilibrium state of a collapsed homopolymer. The polymer is quickly collapsed, crumpled, so created in a configuration that has no knots, and it remains knots-free because you don't allow chain crossing. So you don't allow for topoisomerase activity. If you were to allow this chromosome, this homopolymer to equilibrate, then you will go into the equilibrium globule, which is knotted. So essentially what I'm trying to say is that the ensemble of structures produced by the minimal chromatin model is very similar topologically to the ensemble of structure predicted by the fractal globule, but the mechanism of formation of these two chromosomes, of these structures, is very, very different. One is an equilibrium model, the other is a non-equilibrium model, and I think the fact that you not letting equilibrate by not allowing chain crossing might be problematic because topoisomerases exist. So how am I doing with time? I have half an hour, right?
Yeah, so you're pretty close to done.
Okay, so I'm wrapping up.
Okay.
So so bottom line is we've been looking into, I'm gonna skip all this other part which was there just in case. So the bottom line is that we have a good grasp on what is the physics, what are the physical processes responsible for the folding of chromosomes. So we think there's two main processes. One is phase separation and the other is lengthwise compaction. We have developed a theory to extract this information from experimental data sets. And then we have validated that theory toward other experimental data sets. So we showed that we could predict the structures for a variety of cell lines, and we're now making it sort of like a more of a straightforward tool to be used also by the biology community. We have connected the phase separation, which is observed in the structures, to epigenetic information, which was a major conceptual step. And then we also looked into sort of dynamical features of this energy function, which the energy function was developed to be reproducing the thermodynamics. But we have actually shown that it does fairly well also in reproducing the dynamics of chromatin. And now in this, the part that I'll skip, so the ensembles of chromosomes are fairly fluid, and yet very well defined. And in the past few years, we've been connecting sort of this mechanisms, this physical mechanism of chromatin organization toward evolution and toward the molecules that act as motors or on DNA. So I hope I gave somewhat interesting overview of our work, and I would like to acknowledge all my collaborators. This is a large group, and all this work's been done in close collaboration with CDBP members, Jose, Peter, Eretz, now Tingwu, and also all the wonderful postdocs at the center, my group on the left, and also collaborators from other institutions, like our collaborator in the Netherlands. Yeah, funding is NSF and NIH, and thank you. Am I in time? I can't hear you, Margie.
Margie, you're muted.
Sorry. Okay, there you go. Do you have a question from a student? If so, please unmute yourself and go ahead and ask. Y'all can be brave.
So who's the student?
I don't know. Well, there's Northeastern. Oh, what a nice place to be.
Oh, okay. So there isn't specific.
All right. Well, do we have a question from a non-student?
Yeah, maybe I could ask a question. Michele, very nice talk. So at the end, when you compare the crumpled globule with your new model, you seem to suggest that obviously one is equilibrium, another is non-equilibrium. They are fundamentally different, but you seem to suggest that they are structurally similar. I was a little bit surprised by that. Is that in some very coarse-grained sense, or because they must be different in some way?
No, I mean, they're certainly different in some way, but they're very similar in other ways. So, no, it's a good question. So, in what sense they're similar? So, the figure you see here, you can see this, the coloring. So, these three polymers, right? They're colored from one end to the other. And this equilibrium globule, you see all the colors are mixed. And that's sort of like an example of it's a result of the fact that they're not, right? So what's close in physical space is not necessarily close along the polymer. The two structures you see on the right, they're both unknotted. And the fact that they are knotted means that what's close along the polymer happen to be close in physical space. And that's why you see these patches of color in which you gradually go from blue to red. And you never see blue and red mixed together. So in that sense, both the fractal globule and the microglobule, they will look in that way. So like from far away, you'll see that the coloring, it's in itself a feature of the structure, and they'll give very similar structures. So in that sense, the detail of the structure of the polymer on the smaller scale, they're different. And on that you're correct. But the global organization of the chromosomes, in which you have that what's close along the polymer, it's close along in 3D space, they're similar. So,
but probably the correlation functions and the scaling exponents are different, right?
Yes, but they're much closer than the equilibrium globule, where you have everything mixed, so that the correlation exponents are going to be very different. So Yes, I mean, I'm not claiming that they're exactly the same, but a lot is similar.
Yeah, thank you.
It's
Yeah.
actually interesting because that's a testament to how good was that model. Because, yeah, this is 35 years ago, with the only assumption of the fact that the chromosome should not be knotted. They came out with pretty reasonable architecture, which is
Okay.
very simple assumption, very elegant.
I'm going to interrupt. I'm
Yeah.
sorry. We need to get Evelyn in. So we're
Okay.
way over time. And y'all can talk after if you need to.
Sure.
Okay. All right. So you will unshare your screen so that Evelyn can get her slides up. So now we have Dr. Evelyn Tang, who's going to present. She completed her PhD in physics at MIT in Boston, and then went on to do a postdoctoral fellowship at the University of Pennsylvania. She was a Afric postdoctoral fellow. Then more recently, after that, she was a group leader at the Max Planck Institute for Dynamics and Self-Organization in Germany. And then she came to join CTVP at Rice in 2021, and we are delighted to have her. So Evelyn, when you are ready, you may begin.
Okay. I'm going to be talking about a different kind of topology from what Michele did. Hopefully it will be just as entertaining. What fascinates me is the idea of robustness. So if you look at your brain network and my brain network, they look very different, but yet they can maintain many similar functions. And so we see that even though there's a lot of heterogeneity, or even randomness when you get down to the cellular scale, yet there's a lot of similar functions that can be sustained by these stochastic and heterogeneous parts. And thinking about how these different parts can act together is even more amazing when you think about sort of long time scale or long length scale behavior. For example, we think about circadian cycles and memory, in which the underlying components may have lifetimes or may have timescales, transition timescales that are much faster than these longer timescales. You know, how is it that with all of this randomness and heterogeneity, we get these long, stable timescales? And of course, these are out of equilibrium systems, which, as Michele already alluded to, oftentimes we do need new models that can describe these different regimes. So today I'm going to be discussing mostly some new results that we have on oscillators. And then if there's time, I'll also discuss how these models can also describe dynamics in other molecular systems from like microtubules, or also collective properties of different oscillating systems. And here the theme is self-organization, in the sense that here we're getting emergent phenomena where you have underlying constituents that act, but their overall behavior is different from just adding up what the individual guys do. And here, as theorists, we want to try and predict the resulting emergent functional or dynamical states. So to give an idea of the kind of topology that I'll be talking about today, because that is what sort of unites these different systems, I'm going to just use this example from electronic physics, quantum physics, in which here you have a conductor. It's a two-dimensional conductor, and with an external electric field here and a perpendicular magnetic field, then the electrons can move around and make cyclotron orbits. And in this case, you can see that in the bulk, these cyclotron orbits just move around. They don't go anywhere because the field is pretty strong. And yet on the edges, they bounce off the edges to form these orbits that move in one direction one way and the other direction the other way. What is attractive about these states, and this goes back to the original theme that I introduced, which is that of robustness, which is that somehow these systems manage to remain robust to changes or impurities in the system. So imagine here that if you were to tear the system or deform it so that you get a new edge or some kind of blockage in the system, instead of making these edge currents just fall apart or dissipate back into the system, instead now here, there will be a new edge, and these edge currents will just continue around the new edge that is formed. And so this idea of robustness is very attractive, because then you could have different impurities or different material properties of the system, but yet they wouldn't matter when you're measuring the edge currents, which is an idea of robustness that we want to have similar functions that can be sustained, even though the underlying network can look heterogeneous or there can be randomness in the system. Of course, this electronic system looks somewhat simple and very different from the kinds of biochemical systems that we are used to thinking about, but actually there are analogs in chemical reaction space. One of which is because these orbits or these out of equilibrium cycles, actually, you do see many out of equilibrium cycles in biochemical systems that are often powered by the consumption of ATP or GTP. One example is that of futile cycles, where here you see a phosphorylation, dephosphorylation loop, where it goes right back to where it was, but this consumes energy because to have this cycle, there had to be some input of energy in order to drive it in this going forward and back again. And these futile cycles show up in many different biological systems, and it's not very clear why biological systems would pump so much energy into a system just to bring it back to where it is. However, here we can see already an analogy to the quantum Hall effect that I just showed you before in the previous slide, which is that they can form these orbits that could potentially, then if you link them up into networks, then support these edge currents or these directed trajectories on the edges of the reaction space. So let me show you an example of where we could, and how this could be useful for biology. So the outline that I want to discuss today is as follows. I'm going to show you some of our models, especially we're going to be looking at emergent oscillations and how on the edges of reaction space, we can again get these large system oscillations. We're going to be discussing some new properties that emerge from these unique topological models. And then if there's time, I'm going to discuss the underlying topological formalism and give a couple examples of other biological systems that these models could be relevant to. And feel free to stop me at any given time so that we all stay on the same page. So the model, so this is a system that I'm going to be mainly discussing today. These are hexamers that have 6 components. So ignoring the structure for a while, the main transition that I want to point out is that of these phosphorylation reactions that they can maintain or that they can sustain. So there's 2 different phosphorylation sites that are here shown with this orange little circle and this pink little circle respectively. And so we can get phosphorylation reactions. So here this is phosphorylating in the orange space. This is in the pink space. This is now phosphorylating in the orange space. And we also can have dephosphorylation, where here now you see this pink site gets dephosphorylated, and now here this orange site gets dephosphorylated. So these are loops that are quite similar to the futile cycles that I showed you. And you can see that the number of phosphorylation, the phosphorylation level of both this orange and the pink are given by the numbers in the brackets respectively. So here we get 1 orange and 1 pink, here 2 orange and 1 pink, etc. And this characterizes the state of the system alongside with this internal state that can then tell the system, well, should it be now phosphorylating or dephosphorylating and in which space? So specifically, there are 4 different possibilities that a system can undergo at any given time, and marked by these different labels. So A says now it can undergo a pink phosphorylation, B says it can now undergo an orange phosphorylation, pink dephosphorylation, and so on. And these different internal states can be distinguished by some other processes. For example, this could be, this happened via some conformational transformation of the subunits. So here going from these diamonds to these circles respectively and back, or they could be the binding of some other molecule, etc., which we'll get to later. But here I just want to show that there can also be different transitions between the internal states. So here you see that the phosphorylation level is not changing, but that these A, B, C, D internal states are. And so then for every phosphorylation level, we get these 4 internal states, and then we can lay this out along the space of phosphorylation. So this whole thing can be written as a lattice. So now here, this horizontal axis describes now the phosphorylation increasing in the orange space, and this y-axis now describes the phosphorylation increasing in the pink space. And then we get all of these loops that we described before. So this particular loop that I described up here on the top left describes this loop as we go around in this space. And then this one down here, in which the number is not changing, but the internal states is this one that I'm showing down here. And this is a square lattice where you can see that this unit cell is tiling the space. And the reason why we've described it as in this form is because that this allows us to actually look at the topology and actually the resulting dynamics of the system. So there are 2 different regimes here predicted by topology that come about by comparing the 2 timescales that we have in this system. So the 2 timescales are denoted by this solid and this dashed respectively. The solid ones are changing the phosphorylation level. The dashed ones are not, they're changing the internal state. And because you see that the solid and the dashed are of sort of 2 opposite chiralities, they form loops of different chiralities, when these timescales are similar, you can see that these systems essentially just form a random walk around the state space. They just traverse the space around, and in time they will explore the whole space ergodically. But the other regime happens when these solid of these black rates are faster than these gray rates. And in that case, the system, once it hits the edge, it's going to stay on the edge, as you can see by inspection. So let's say we're here. Here, the system can only move to the right. Here, if this is faster than the gray, it's going to move there. And here it can only move to the right. Here, this is more likely because it's faster. It has to move here. It has to turn a corner. Now this is more likely. It has to move here. And so in time, the steady state is going to just stay on the edge. And this is what we saw happen in our electronic example, in which here, what we saw is that although there was the possibility to make loops in the center of the system, but what really dominated and what could be measured at any given time was this cycle that goes around instead the edge of the system, and that is the main dynamics that you see in this system. So and this topological model also has this property of robustness that I was mentioning, which is that now here it's going to go around the steady state is going to go around the edge. But then now let's say if we have blockages or some kind of inaccessible states, then here, instead of just dissipating this edge state or making it fall back into the system, then here you can see that, let's say if this is inaccessible, then the system now here will just round the corner up here. This is more likely. Now this is more likely, and it's going to move here and turn the corner, so that in time, it's just going to still go around the edge, whatever the new edge is, and maximize this edge. So this robustness, again, seems like a desirable property for a system that should be able to withstand different changes in the environment or different stimuli that come into the system in order to achieve its goals. So we can now compare it to see whether this could be something that describes dynamics that we see in biological systems. And that's what I'm going to do next. But I just want to pause and see if there's any questions so far about the model before we move on. One property I should point out is that here you see that the main ingredients is this difference of time scale. So this gray versus this black, and that definitely can tolerate some amount of disorder or difference. But so long as that is in place, then we do get these edge states, which is also something that's nice that can allow us to then test or compare to real systems.
I've got one question.
Yes.
So I see that the final state would be to go around the edge, but I don't see how you would have got this graph to begin with.
So the graph is inspired from topology, which is very well studied in quantum electronic systems. They have all kinds of models that show different edge properties. That was my PhD was on that. So, once you do a PhD, then you start getting an idea of these models. And essentially what we've done recently is to then come up with a description for topological systems of this sort in a stochastic framework. Did that answer your question?
I think so, and I think you're probably going to answer it more in a second.
Yeah, and if you're asking instead about, what does this have to do with any actual biochemistry, then that's what I'm
right. I think that's what I'm trying to do. Yeah, I think you're about to answer it.
Yes, perfect.
Thank you.
Anything else? Any other question? Okay, great. So let me move on, since there's interest in how this relates to biology. Now I'm going to discuss how this could relate to a biological oscillator. There's tons of biological oscillators describing cycles of different lengths and kinds, one of which is a very sort of nicely isolated and better understood system is that of Chi-ABC, which regulates the circadian rhythm of cyanobacteria. It is a hexamer, so, with 6 monomers, which is gives you a clue of why I had drawn the molecules like that previously. And to regulate this day-night cycle, what happens is that on each monomer, there's 2 phosphorylation sites, the T and the S respectively. And what happens during day is that all of the 6 T sites get phosphorylated, and all of the 6 S sites get phosphorylated, and then all of the T sites get dephosphorylated, and then all of the 6 S sites get dephosphorylated. There's also other changes. You can see there's a conformational change here of these A loops that are sticking out in these cases, and here they're sort of buried versus exposed. There are also binding of other molecules, like in this case, chi A is binding, here is unbinding, and then chi B is also binding in some cases. And so overall, this creates this long 24-hour rhythm. However, so this is just what I was explaining. However, one question is, why is this global cycle so robust and so concerted? And the reason why this is surprising is because if you look at, or if you think about what the individual monomers do within the hexamers, you see that there's a lot of fluctuations, a lot of mixing, shuffling. Presumably, the monomers can undergo their own individual phosphorylation, dephosphorylation cycles. The monomers can bind into hexamers and unbind into individual monomers. So monomers shuffle between different hexamers at different times. That was shown very early on, sorry, and also even within the same hexamer, these A loops can take on different conformations. Sometimes they can be exposed and sometimes they can be buried on across different monomers within the same hexamer. So it looks like monomers can do a lot of individual transitions themselves. So why is it that they would then all phosphorylate T sites at the same time, and then all phosphorylate S sites at the same time, and then all dephosphorylate together also? I mean, obviously this sustains this global cycle, but what is the mechanism that then makes them all do this together, despite a lot of what presumably is a lot of individual stochasticity and movements also? So to try and answer this, we propose a model that, we propose a model that essentially is using this topological model that we had shown before. So now here, this orange and pink phosphorylations are described as T site and S site phosphorylations respectively. And these internal states can now be, again, going from these exposed loops to these buried A loops, as well as the binding of chi B and chi A respectively. And now we also allow for both the backwards transitions in addition to the forwards transitions, which we denote by a prime compared to the original unprimed labeling. And all of this is the same model that I showed before. Now here, the horizontal axis describes T phosphorylation and the vertical axis describes S phosphorylation. And this model goes from 0, 0 to 7 by 7, describing the full state space of all 6 monomers in the hexamer. And this being the same model that I described before, so long as now these black rates, especially they still have a dominant direction. So long as these black rates are still faster than these gray rates, then the system will still behave in the topological regime where these black rates are faster than these gray rates. The system will still maintain these edge states so that they will now undergo all of the monomers are undergoing T phosphorylation, all undergoing S phosphorylation, all undergoing T dephosphorylation, and all undergoing S dephosphorylation. And these are similar dynamics as what has been proposed by other established models in the literature, except that here in our case, we didn't make any assumptions on how the bulk states compare to the edge states. Here, the transitions, we have the same strength of transitions in the bulk as in the edge. So we still allow for the individual monomers to have their own transitions. But here, because of the topology of the network, then this supports this global edge state, unlike in oftentimes in other models in which we have to impose that the external rates are stronger than these sort of these edge rates are stronger than these bulk rates by hand. Here you can show that they're depicted as being negligible, also by invoking other pretty strong assumptions about an all or nothing mechanism, which is why that the system has to change altogether before then it dephosphorylates. And we do know that there are observations, given that monomers don't always behave as all or nothing, that they can have exposed and buried loops in the same hexamer, etc., that suggests that these assumptions are a bit too restricted. So we are now in the process of characterizing what new properties these topological models have for oscillators. And we are looking at things like the coherence. So how do these oscillations do in terms of being stable and coherent? And we're comparing that with other models. And one thing that I want to show you quickly here in the interest of time is that we are getting a very interesting regime in which coherence is normally increased by increasing the driving, the thermodynamic driving of the system. And instead, in our system here, what we have is a regime in which, even though we have increased thermodynamic driving, we actually have decreased entropy production and a decreased cost to sustain these more coherent oscillations. So let me explain how this could work. And that's because as we increase the thermodynamic driving, where here thermodynamic driving is defined as this ratio of the forwards to the backwards rates. As this driving increases, then the dynamics become localized on the edge of the system. They go from this random walk here or being sort of homogeneous to now being localized on the edge, and hence also the entropy production becomes now localized on the edge. And so actually, when we now look at the total entropy production, we see that it has two contributions now from the edge, the sigma E, as well as from the bulk, the sigma B. And so while we are increasing in the driving, we see that the edge entropy production increases as we expect, because now there's more dynamics on the edge. But then there is a regime in which the bulk actually drops quite precipitously. And that's because there's so many more bulk states than there are edge states that, because they're decreasing, that the number overall, that the decrease is actually much stronger than this increase in the edge. And so the total entropy production decreases, which leads to an interesting regime that here we're actually increasing in coherence while decreasing in the cost or in the cost needed to sustain these coherence oscillations. So this is something that we are studying a lot more in the group. And but here now I just wanted to now step out and then to discuss a little bit more, what are some of these topological properties of the system, as well as show some other dynamics that these models can exhibit. Are there any questions before now I move on to this last part of my talk? Okay, great. So here I just want to illustrate that we are using the master equation and Markovian dynamics to describe these systems. And essentially we're using a mapping of this master equation that looks very similar to the Schrodinger equation up to an I, to use some of the results that were obtained for quantum Hamiltonians to look at the topological invariance of this operator, W, where they actually have identical eigenvectors as the corresponding Hamiltonian, so that in lattice systems, we can actually look in Fourier space at the winding of the eigenvectors, and then we can integrate these eigenvectors to show either you get a trivial state in which there is no particular preference of the system to spend time on the edge versus the bulk, or the system can be topological, where the system prefers to spend its time in the edge. We can do simulations and show that this is indeed the case, where here in this 2D plane, and where this is time, the system after a while starts to spend time on the edge, so that the steady state is indeed on the edge. And we can also start now to look at the properties of these edge states to see that they are exponentially localized, so that even for small systems, they do not blur into each other, so that for even fairly small systems, these edge states can be robust. I'm just going to take the last few minutes now to just show a couple of other examples that we can get. We also have a different model where now, instead of just conversion or phosphorylation, that here now phosphorylation can be thought of as adding these little squares, but also one of which is the second, where the number of squares now is the second number in the bracket. But we can also have a different process, such as growing and shrinking, and that can describe the first, that is described by these green blocks and the first number in the bracket. So here you see that the system now has this addition of a molecule, some kind of conversion of phosphorylation. Now this grows, and then now it shrinks, removing a converted molecule. So this is now a 3-state system in comparison to the 4-state system that we had. And we can also have internal states in this system, that different internal states that show the system what's going to happen next, as well as transitions between these internal states. And this can be laid out as a Kagome lattice here in this case, that also shows edge states around this triangle, so the steady state and the edge state properties. So this is useful to describe other types of systems, and in particular here, if we want to describe a system that's growing, let's say on this axis, and then converting and then shrinking. And we can also do different things by, let's say, instead of just having two types of rates, the black and the gray, we can change one of the rates. So let's say here we change always the upper right rate to make it now in between these two rates, so that it's a little bit faster than these rates, but not as fast as these black rates. What happens here is that we see that now the system is going to grow more slowly. This is going to convert quickly and shrink more slowly. So if we just plot the growing and the shrinking, we do see that this grows and shrinks as we expect, converting pretty quickly. But sometimes also we see that it doesn't make it all the way to the edge. So sometimes it cuts out and makes these smaller triangles, which here we can see that this is reminiscent of this sort of abrupt, this dynamic instability of microtubules in which they don't always grow to the same length before abruptly decaying. So we are also studying that. And the last example I want to show is that we can now couple systems together, because let's say these systems can share the same constraints. Let's say if they're both growing and binding to monomers, if there's a common pool of monomers, then this actually provides a constraint that they both have to compete for this pool of monomers. So here we have one system that's growing from the left towards the center, one growing from the right towards the center. Once they meet, they'll have consumed all the available monomers in the pool, and they have to move to the next reaction. And so then they start to move up. And then you can see that after a while, because of this competition, they start to go one goes up at a time. And so they form this anti-phase synchronization. And we can also obtain synchronization by looking at other places in the parameter space. And now we're looking at increasing the number of systems and the number of couplings to see what behaviors we can get. So this is what I wanted to say. I'm just going to step out and say that here we are looking at non-equilibrium systems, because now here W is non-symmetric. And so now we get lots of new and exciting properties. And this is the summary of what I was telling you about. Here we have a topological framework that can describe different kinds of behaviors, clocks, growth and shrinkage and synchronization. And this is a broad framework that can be describing many different scenarios. This is some different types of work that my group works on. So I was telling you about topological cycles. We've also worked on learning and brain networks, as well as flow fields and fluctuation theorems. So some of these other examples are that we think about learning and information. So how fast learners learn, how information propagates in flows, or how navigation can happen on curved surfaces. So I want to thank my, fledgling group. So my student Changbin was the one who's been exploring these different contributions of entry production and coherence and other properties. Alexandra's a postdoc in my group. Here are some of my collaborators when I was at the Max Planck. And of course, we are still hiring as a fairly new group. And I want to thank all of you, and I'm happy to take questions. And let me put up the summary that I had before.
Thank you so much. That was really interesting.
Great. I'm glad you liked it, Margie.
Yeah. Do we have a student question?
Hi there.
Go for it.
So I guess I have a quick question. Based on the fact that the difference in the rates of the bulk states versus the edge states is important to have it preferentially move along the edge cycle, how does having higher rates in the bulk states influence the time delay, I guess, to make it to the edge and start going around the edge cycle?
So I just want to clarify that that's actually not what we have. We actually, so let me just show. So actually, so you were saying, like, do I have different bulk states versus edge states? And actually the answer is no. And this is actually exactly sort of the new feature of our model, that you see that we're just repeating at every single time this same motif across the entire space. So there's no difference between the bulk and the edge. And in fact, the reason why the system prefers staying on the edge is because of the topology of the particular way in which these transitions happen. So this is actually a completely different way to get the system to favor the edge versus the bulk, as compared to other models that I was showing you, where here often this difference between the bulk and the edge is put in by hand. Instead, for our case, there is no difference between the bulk and the edge states in terms of their magnitude.
So
yeah, go ahead.
I guess my question was, if you're on one of the cycles with the dark black loops, because
Yes.
the dark black arrows have a faster rate, how long do you stay on one of those cycles before you move to the edge and then remain on the edge?
Yeah, so that is a steady state question. So yes, one can definitely get caught in those cycles, but you can imagine that a lot of these transitions happen extremely quickly. There's, maybe thousands of them at any given in every given second. And so if you found that at some point you're going to escape and you will start to explore the space. And so yes, you will get transients in which you get stuck. And but in the long time limit, you will go on the edge. And yes, there will also be transients in which you sort of unbind from the edge, get stuck again in the bulk, but then you will come out and you will end up spending most of the time. So obviously, the more time that's spent on the edge versus the bulk increases as the black is larger than the gray. If the black is larger than the gray by a little bit, it'll spend just a bit more time on the edge. But if the black is very much larger than the gray, then it's going to spend in the long time limit most of its time on the edge.
Okay, thank you.
Great question.
Any other questions? All right. Well, thank you all for coming to the return to the iPoLS faculty seminars. We'll have some student seminars and some career panel discussions kind of in rotation throughout the rest of the year. So you will be getting more emails from me in the future. And thanks so much to our speakers, Dr. Di Pierro and Dr. Tang. This was great. Thank y'all.
Thank you, Margie. Thanks,